This procedure applies a combined EEG and near-infrared spectroscopy recording technique to determine to what extent decreased neurocognitive performance in weightlessness is due to the primary effects of hemodynamic and electrocortical changes or to secondary stress-related effects. This is accomplished by monitoring changes in electrocortical activity of participants during a parabolic flight, including phases of microgravity, hypergravity, and normal gravity. Hemodynamic changes within the frontal brain can be monitored in parallel using near-infrared spectroscopy, or NIRS. The changes in brain cortical activity can then be localized using electromagnetic tomography. The final step is to correlate the electrocortical and hemodynamic changes. Ultimately, results show that the hemodynamic changes due to changed gravity conditions are associated with changes in electrocortical function.
The main advantage of this technique over existing methods like MRI or PET is that electroencephalography combined with electrotomography and near-infrared spectroscopy is feasible during extreme environments like parabolic flights or space flights with hypergravity, weightlessness, limited loads, and limited space. And it allows to measure and correlate changes in neuroelectrical activity and hemodynamical changes within the brain.
This method helps answering key questions in the field of neurophysiology and space research, such as changed gravity conditions leading to hemodynamic and electrocortical changes. It helps answering where exactly in the brain these changes happen and what the consequences of these changes are.
Once we have identified the underlying neurophysiological processes, once we have identified what happens